The higher the GC content, the higher the stability of the DNA. So that is why we have what we call a melting temperature. A melting temperature is a temperature that can denature the molecule of the DNA. We have, remember we said we have double-stranded DNA. So DNA is double-stranded like this. So we have a melting temperature. A melting temperature is a temperature in which the double-stranded DNA can be opened up. It will be denatured. So that's what you call melting temperature. And the higher the GC content, the higher the melting temperature. What is GC content? G means guanine and C means cytosine. So the higher the GC content, why is it that the higher the GC content, the higher the stability of the DNA? Is because, remember, between G and C it is 3 hydrogen bonding. So the higher the hydrogen bonding, then the stronger the molecule it is and the higher the stability. Do you also get this point? Do you get this point? Melting temperature, GC content, and then stability of the DNA, they are related. Melting temperature, it is a temperature that denatures the double-stranded DNA into single-stranded. So it opens off the DNA molecules from it is double-stranded into single-stranded. That is what we call melting temperature. And we also have GC content. So usually the higher the GC content, the higher the stability of the DNA. Why is it that the higher the GC content, the higher the stability? Is because the G and C fare through 3 hydrogen bonding. So the higher the hydrogen bonding, the more the stronger the molecules is. Please, is that clear? Yes, the GC bond is triple. Yeah, it's triple. And the higher it is, the higher the stability of a molecule. Please, do you understand this? Okay. So then the next thing, ladies and gentlemen, please, do you have any question regarding the DNA and RNA? I know that I sent you the video yesterday. So if you have any question, please ask. Ask me any question you have regarding the DNA and RNA before we move to carbohydrates. If you have any question regarding the DNA and RNA, that is nucleic acid, please ask. I send you 2 videos on nucleotides and the DNA molecule. So please, do you have any question? So if you have any question, please ask on the comment section so that at least we'll address it. So please explain the melting temperature again. A melting temperature is a temperature. A melting temperature is a temperature that denatures the DNA molecules from double-stranded into single-stranded. I saw questions that were asked last year related to this melting temperature. So it is a temperature that opens up the double-stranded DNA into single-stranded. And the higher the GC content, the higher the melting temperature and the more the stability or the more stable the DNA is. Please, do you have any question again?